Good morning. Hi, everyone. So it looks like we might have kind of a light crowd today, Friday morning, 8 o'clock, BIO101a. So a couple of reminders. We changed office hours, so remember that there are no office hours today. If you want to come to my office hours, they're now Tuesday and Wednesday, 9 to 10 a.m. in Stanley Hall 482. It's posted on BSpace if you forget, but just that's a reminder next week, Tuesday, Wednesday. Also, I'm getting a few emails from people. I don't understand this and that from the lecture. Please, here's a tip. When you're reviewing your notes after lecture, the first thing to do is go to your book and read the reading that is corresponding to that lecture. And I always post that on the first page of the notes and the slides. Read that first, because a lot of times you'll find that your questions are answered very nicely by the book. If you read the book and it still doesn't make sense, you can email, you can go to your GSI office hours, you can go to Mike, you can come to my office hours. Okay, so lots of ways to get help if it doesn't make sense, but please first try to read the book and see if it clarifies any questions that you might have. Okay, so today we're going to talk about biological membranes. And this is a discussion this is basically material out of chapter 7 in your book. Again, a very big area of biology and something that many laboratories have been investigating for a long time. So we will really try to touch on what are probably the most important aspects of membranes, how they're put together and how they work. And I'm going to tell you four things. We're going to talk about the current understanding of how membranes around cells actually work. We're going to talk about the kinds of proteins that are found in membranes. And then we're going to talk about the ways that molecules get in and out of cells, the way they get across membranes. So this is kind of the overview. So the plasma membrane, we've talked about this before, right? We talked about phospholipids, the lipids that make up most cellular membranes. This is the boundary that separates cells from the outside world. And the plasma membrane is very important because it has selective permeability to molecules. That means that molecules can't get freely inside and outside of cells. They have to go across the plasma membrane. And as we'll talk about today, there are a number of different ways that they do that. Does anybody know what that is? First of all, what kind of a molecule is that? Is it a lipid? Protein. I heard somebody say protein. That's right. And anybody know what it is? It's a channel protein. Yeah, so this is actually a potassium channel, and this is incredibly beautiful structure determined by crystallography, and we're actually looking at this protein as it's embedded in the membrane on either side. So here's the channel right in the center where individual potassium ions can go across the membrane. Right? This is a structure solved by Rod McKinnon at Rockefeller University a few years back, won him the Nobel Prize. It's incredibly important in biology for nerve transmission, for example, and we'll talk a little bit more about these kinds of proteins today. Okay, so, but I first want to talk about the way that membranes are put together and how we currently think that they're structured. So this is a slide that should look kind of familiar to you, right, because we've been talking about phospholipids, and so you know already from what we've talked about that these phospholipids are made up of a glycerol backbone that has a phosphate group on one end and then two fatty acid chains that are esterified to the other two oxygens on that glycerol. This is all from lecture 2. And those fatty acid chains are hydrophobic, and so they tend to want to come together in the center of the membrane, whereas the phosphate head groups are hydrophilic, meaning they like to interact with water, so they tend to be on the outside of the membrane. And so that allows the membrane to come together in this kind of a bilayer structure where we have phosphates on the outside and then these fatty acid chains on the inside. And you might also remember that we talked about the different levels of saturation of those fatty acid chains. If they're more highly saturated, meaning they have more hydrogens on their carbons, they tend to be straighter, right? And they tend to stick together better at lower temperature. right? If they have unsaturated sites, they have less hydrogens, they have some double bonds, and then they tend to have bends in the fatty acid chains that allow them to, don't allow them to pack together as well at a lower temperature, right? So that's an important principle to know. So these are the most abundant lipids in the plasma membrane, and they have a property that it's important to know that these are called amphipathic molecules, and that means that they have both hydrophobic and hydrophilic regions. Okay, so amphipathic is a word that we also use sometimes to refer to proteins that have hydrophobic and hydrophilic properties. So you'll see that word in your book, and it's an important one to know. The fluid mosaic model is the current model that scientists have for understanding how membranes are organized. And this is a model that basically says that a membrane is effectively a fluid structure that has a mosaic of various proteins that are embedded within it. And so when we think about the way that a membrane is organized, we don't think about it as something that is staying the same all the time. It's in fact, all those molecules that are in the membrane are quite mobile. And I'll show you a couple of pieces of data that I think experiments that have been done that really show this, I think, pretty nicely. Okay, so this is kind of a closer view of the lipid bilayer. Now we're looking at, again, at sort of an artist's rendition of what a section out of a membrane would look like. And you can see that we have all these phospholipids packed together very closely, but then every now and then there's a protein, and we talked about this a little bit last time, right, that there are proteins that will span the plasma membrane, and they tend to have hydrophilic regions that are protruding either inside or outside of the cell where there's an aqueous environment, whereas the interior of these proteins that's packed in the middle of the lipid bilayer tends to be hydrophobic, right? So these are amphipathic molecules. They have both hydrophobic and hydrophilic properties to them. There's a little bit of an interesting history here that I just wanted to mention to you briefly. So for a long time, cell biologists have been studying cells and membranes for a long time, basically ever since microscopes were developed. And it became pretty clear that cells were surrounded by a barrier, and people were trying to understand what that barrier was and how it was put together. And so for a long time, really up until through the 1960s, the idea was that the membrane was in fact a sandwich that had lipids in the middle of it, and it had proteins on either side. And this was called the sandwich model. And it was really based on the fact that in low-resolution light micrographs of cells, it looked as though the plasma membrane had 3 layers to it. But then in the late 1960s, a lot of people began to realize that the sandwich model for membrane structure really didn't make sense in a couple of important ways. So it didn't explain how proteins could have, and people at that point had started to recognize that some proteins could be hydrophobic. How could they be sticking on the outside of the membrane? They would not want to do that in general. And also, as microscopy got better and better, it was possible to look at higher resolution at membranes, and a number of people noticed that the membranes around various organelles, like around mitochondria, for example, didn't look like they had 3 layers to them. They looked like they had 2 layers to them. And so eventually, this was resolved by some very nice work that was done in the early 1970s to propose the current model, the fluid mosaic model. And now what we know is that the plasma membrane looks like it has 3 layers to it, because what we're seeing at low resolution is we're seeing the bilayer, the lipid bilayer, but we're also seeing a 3rd layer of extracellular matrix, those proteins that we talked about last time that are protruding outside of the cell. They're not on the mitochondrial membrane, right? It doesn't have those same proteins on the outside, so those membranes look different. So anyway, in the early 70s, the fluid mosaic model was proposed to explain the way membranes are structured. And one of the things that's really cool in science and in biology is that scientists will come up with what we call models to explain experimental data. And then those models serve as a platform for doing further experiments. So once the fluid mosaic model had been proposed, and it seemed like it explained a number of observations, many laboratories busily set about testing it to figure out, does this really make sense? Is this really true? And so there have been a couple of experiments that have been done that I wanted to just share with you that are in your book and are really interesting because I think they test this model and they so far have shown it to probably be right. And one is based on a technique in microscopy that's called freeze fracture. And what you do in a freeze fracture experiment is you can take a cell or a tissue and you can freeze it in such a way that you can use a very thin knife to peel away part of the plasma membrane. So basically cutting that membrane in half and peeling it back. And here's a drawing that sort of illustrates effectively what is done in this method. And when you do that, you can peel these layers back and then have a look by using microscopy to figure out what you see going on in between the layers of the plasma membrane. And when you do that, what people have observed is that you see inside the cytoplasmic layer this sort of studded surface that we think represents this surface right here, where we've got these proteins that are protruding through the membrane because they're embedded in the membrane. And then if we look inside of the extracellular layer, we see a surface like this, a smoother surface that represents the outside where there are little bumps of proteins coming through, but they're not as prominent. Okay, so it really suggests that's the way that membranes are set up with these proteins that are studded through the membrane. One of the important aspects of this model is that these membranes are fluid. So the idea is that these proteins are not static in the membrane, but they're actually moving around. They're diffusing around the membrane, and that's very important for the function of the cell in many ways. So you could imagine that some of these membrane proteins, for example, are receptors. They have to be available to bind to molecules, and those molecules have different locations on the outside of the cell. They need to be able to move around to access and interact with the kinds of molecules that they bind to. Now this slide right here illustrates a couple of things. So first of all, that these phospholipids themselves in the plasma membrane can move around in the bilayer. This is kind of an amazing number, actually. So this shows you here that we think that the lateral movement of these molecules, these phospholipids in the membrane, is occurring about 10 to the 7th times, about 10 million times per second. And so that means that an individual phospholipid could move from one end to the other of a typical bacterial cell in a second. So these guys are really moving, right? Really fast. And in contrast, flip-flopping, so moving across the membrane, occurs very slowly. So this fluid, the idea of a fluid membrane is really occurring mostly in the horizontal direction, right? Molecules are moving horizontally. And most of the lipids and proteins can drift in that direction. And then again, this is showing you what happens if you have, depending on the different levels of saturation of these fatty acid chains, we can have a membrane that is more viscous like this with more saturated fatty acid chains, or we can have a membrane that is more fluid because the fatty acid chains are less saturated. And that'll depend on, so if you have, if we looked at the plasma membrane from bacteria growing in the Arctic, we would tend to find fatty acid chains that are, what do you think, more or less saturated? I heard more and less. Less. Yeah, probably less, right? Because in general, we want these membranes to be more fluid at lower temperature for those cells, right? And then down here, this is kind of important. So we talked a little bit last time about cholesterol. We talked about cholesterol being an essential component of animal cell membranes. And you might have wondered, well, why is that? And actually, it's in part at least because these cholesterol molecules embed themselves within the membrane, and they help control the fluidity of the membrane because they actually have this effect of, you can see they sort of break up these fatty acid chains, and would probably in general tend to cause these membranes to be more fluid in general at a lower temperature. Okay, and then here's another really interesting experiment. So this was done, again, back in the 70s by Michael Edidine at Johns Hopkins and his colleagues. And the experiment they did was the following. So they took mouse cells and they labeled membrane proteins on the mouse cell with one type of dye. And then they took human cells and labeled membrane proteins on the human cell with a different type of dye. And then they fused these cells to form a hybrid cell. And when they did that, the experiment then was to ask what happens to these proteins. Do they all stay localized on the surface where they first started off, or do they move around in the membrane? And so they waited a while and went back and looked, and actually after just about an hour, they noticed that these membrane proteins were now very much intermixed on the surface of this hybrid cell. Beautiful experiment that really showed very nicely this, illustrated this idea of the fluidity of the membrane. And so I think is a piece of data that really supports the idea that membranes are fluid. Okay, and so let's talk now a little bit about membrane proteins and what they do. So these proteins are bound to the surface of the membrane, peripheral proteins. And so if we see the word peripheral protein, we know that we're talking about not a protein that is embedded in the membrane necessarily, but it's really just stuck or interacting to the surface of the membrane. And in contrast, if you see the word integral protein or integral membrane protein, these are proteins that actually pass through the hydrophobic core of the membrane. And so if you think about it, as soon as you see the word integral membrane protein, right away you know that most likely, almost certainly, that protein has to have some hydrophobic regions to it, right, that would allow it to interact favorably with the hydrophobic center of the membrane. For a peripheral protein, that might not have to be true. It might just be sticking to the outer surface. Integral proteins that actually go all the way across the membrane are called transmembrane proteins. That's sort of an obvious word, I guess. Goes all the way across. And this is important here. So the hydrophobic regions of an integral membrane protein consist of one or more stretches of nonpolar amino acids. And very typically, those stretches of nonpolar amino acids are folded up into alpha helices. And so you'll see illustrations in your book that show coils of alpha helical segments of proteins going across membranes. And that's not just totally made up by the artist. That's actually representative of the way that many integral membrane proteins and transmembrane proteins are structured. So one thing that you would want to be able to do, for example, is, if I gave you a sequence of a polypeptide, and I said, first of all, tell me, do you think this is likely to be an integral membrane protein or not? You would want to be able to look at that sequence and figure out whether, is there a stretch of nonpolar amino acids there that would be able to interact favorably with the interior of the membrane, right? And if I asked you, where is that segment that you think, what would you, if you were predicting which region is likely to be inserted in the membrane, you'd want to be able to pick that out. Okay, and that's just based on all the things we've already been talking about, the properties of amino acids and the fact that hydrophobic molecules tend to interact with each other and exclude water, and hydrophilic molecules tend to want to interact with water. Okay, so we can sort of boil down the functions of membrane proteins into 6 categories, and they're listed here. So a very important category, and we're going to talk a lot more about this, is transport of molecules across both ways, into and out of the cell. Enzymatic activity, so certainly not all, but some membrane proteins are actually enzymes. They have the ability to catalyze reactions. They're important for signal transduction. That means helping the cell to figure out how to respond to its environment, often through chemical changes that trigger a cascade of reactions in the cell. They're important for cell-cell recognition. How does a cell know where it is and whether it's in a foreign environment or not, and how do cells recognize foreign invaders that might come in and then try to defend against them? And a lot of that comes about through the properties and behavior of membrane proteins. Intercellular joining, so we talked last time about gap junctions, tight junctions, for example. That's all carried out by membrane proteins that interact with each other on the surface of cells. And then finally, it's membrane proteins that allow cells to interact with the cytoskeleton and also with the extracellular matrix. Okay? And then here's here's the nice picture that shows, sort of illustrates a typical transmembrane protein. It has, so remember that proteins have an amino terminus and they have a carboxyl terminus, right? So they're directional polypeptides. So here's the amino terminus here, and you can see that we've got an alpha helix that's going across the membrane, and then we've got these loops down here. We've got another helix, and then these helices are all connected by loops. And so this is very characteristic of transmembrane proteins. They often have multiple alpha helical regions that go across the membrane, and they can go across the membrane many times. And then these loops are typically, what would you say, hydrophobic or hydrophilic? Hydrophilic, right? Because they're interacting with the water outside or inside of the cell. And so it's often, there now are nice computer programs that allow you to, you can take even a very large protein sequence and you can ask, is this likely to be a transmembrane protein? Even if you don't know anything about it biochemically, or where you haven't seen where it's located in the cell. And often those programs can predict pretty accurately whether it's a transmembrane protein because of that kind of property. Okay, so this slide here is sort of summarizes these different functions that we've been talking about, and we can just summarize these kind of briefly, and we'll talk more about this, a couple of these aspects in a little bit more detail. But this is showing you a couple of proteins that, and the way they work for transport. We can have proteins that form channels that allow molecules to get in or out of cells in a sort of a passive way, but we can also have channels that function through a mechanism that requires energy input through the hydrolysis of ATP, right? So these channels can be passive or they can be active. Again, we said that enzymes can be embedded in membranes, and the cool thing there is that sometimes what we find is that these enzymes are actually embedded. You have a set of different enzymes that are embedded in a membrane next to each other that form a little pathway, a cascade, so that a molecule that comes in and interacts with the first enzyme, and this enzyme makes a product, and that product comes off, it interacts with the next enzyme, and this enzyme makes a second product, et cetera. And so you can get very efficient catalysis of a whole set of reactions by having these enzymes all together in the membrane. Signal transduction, so again, the idea here is that some molecule on the outside could interact with a protein and then transmit a signal across the membrane, and that's often done through chemical or structural changes in the protein itself that trigger the transmission of that signal and then changes inside of the cell. Cell-cell recognition is often a feature of these glycoproteins that we talked about last time. These are proteins that have carbohydrate on their surface, and depending on the cell type, the tissue type, those carbohydrates are different. And so cells can use that information to figure out if it's a liver cell, is it a bacterial cell, is it self, is it non-self. Intercellular joining, so these would be, again, what would occur in junctions like gap junctions or tight junctions where we have very tight binding between adjacent membranes through interactions of their proteins. And then finally, these membrane proteins are important for interaction and attachment to both the cytoskeleton on the inside of the cell and to the extracellular matrix on the outside of the cell. So, these proteins do a lot, and that probably explains why there's been just tremendous interest in biology and understanding how membrane proteins work. And one of the many exciting developments in biochemistry and structural biology in recent years is that more and more of these membrane proteins are being studied in detail. We have increasing numbers of detailed structures solved by X-ray crystallography of these proteins. And so we have a lot more information than we did even just a couple of years ago about how many of these proteins actually work. So membranes have a sidedness to them. So they have a distinct inside and an outside. And it's this asymmetrical distribution of proteins and lipids and the associated carbohydrates that gives it that sidedness. You want to have receptor proteins on the outside of the cell, for example, and different kinds of proteins on the inside of the cell or spanning the cell. And how does that get established? Well, it turns out that really gets established when the membrane is built by the endoplasmic reticulum, the ER, and the Golgi apparatus. And I think the next slide sort of illustrates this process of synthesis, where we saw this in an earlier lecture, but the endoplasmic reticulum is a site where there's lots of synthesis going on. These vesicles are being pinched off. They're fusing with the Golgi. And then the Golgi is, as we said before, it's kind of a storage and shipping and receiving site in the cell. It's a place where molecules are stored for a while. When they're needed, they're then packaged up and shipped out. And when they get shipped out, they're shipped out in these vesicles, and many of these vesicles will then eventually fuse with the plasma membrane. They might secrete things out into the medium, like in this example here, but they also have proteins embedded in them that establish this asymmetry on the surface of the cell. Okay, and so this brings us to this very important aspect of membranes, which is the selective permeability of cells. They can't let molecules in and out randomly. Instead, it's very important that these membranes control the access of molecules to the inside of the cell and help the cell secrete those molecules that at the right time and the right place that are necessary for it to interact with its environment. And so these plasma membranes are selectively permeable. And so this is kind of important to know down here. So very hydrophobic molecules, nonpolar molecules, these would be things like hydrocarbons, also including, very importantly, carbon dioxide or oxygen, right? Those are both nonpolar molecules, they're very small. They can actually dissolve in the lipid bilayer and pass very readily through the membrane. And in contrast, if we have very polar molecules like sugars, they don't go across the membrane easily. They have to be transported in some way, okay? And same thing for ions that are charged. They have to be transported across the membrane. And so I want to talk a little bit about the different ways that these molecules get across. And first we want to talk a little bit about a process called passive transport. And passive transport is really just means diffusion across a membrane with no investment of energy. So the cell doesn't have to burn ATP to do this. It just has to set up a mechanism for these cells to get across, and then they'll go by the process of diffusion. So I think probably most of you are familiar with this idea of diffusion, that this is a tendency for molecules to spread out evenly into available space. And even though molecules move randomly, they will tend to move in the direction in which there's a lower concentration of those molecules. And this sets up a process eventually, if there's equilibrium in a closed system, we have a dynamic situation where molecules are moving around, but the concentration of them across the medium is essentially the same. And I think there's a slide here to show this. Yeah. So this is a slide that shows how this process works. So if we had a situation set up, and this could be a cell, or it could be an experiment you're doing in the laboratory with a beaker that has a membrane in it, and we have water on one side, and then we have water containing some dye molecules on the other side, and we've got a membrane in the middle that allows free diffusion of these molecules across the membrane. We're going to see a net diffusion of these dye molecules over to the side of the vessel that has a lower concentration, or initially no dye, right? And that process will occur until there's an equilibrium established such that there's an equal concentration of these molecules on either side of the membrane, at which point molecules are still moving back and forth, but they're moving such that the concentration is the same on either side. And this could happen also if we have two different molecules that are dissolved on either side of the membrane that are, again, diffusing across, and eventually the concentration of both will be the same on either side of the membrane. Okay, so I think that if that doesn't, this should probably be very familiar to most of you. If it's not, there's a nice detailed discussion of this in your book. And so this then gets us to the idea of osmosis. So this is the diffusion of water across a selectively permeable membrane, such as the membrane of a cell, for example. And so water will diffuse across a membrane from a region where there's a lower concentration of solute to the region where there's a higher solute concentration or higher salt concentration, for example. Okay, and why do we care about this? Well, it turns out to be very important that cells regulate the access of water and small molecules across the membrane because of this property of osmosis. And the reason is that if water and salts weren't regulated, then we would have a situation where cells could be very susceptible to either bursting or just shriveling up, depending on the environment that they found themselves in, right? And so that would be a very bad thing. And so cells instead want to be very careful about controlling how water gets in and out of cells. And so this is just, again, a sort of an experiment that one can do in the laboratory. And if you set up a situation where you have a solution that is connected in the middle by a membrane that, in this case, is only permeable to water and not to these dissolved solute, which in this example is sugar molecules, then what we find is that if we have a higher concentration of sugar molecules on this side of the vessel than on this side of the vessel, water will tend to migrate from this side to this side, right? And it will do so until the concentration of sugar is now the same on both sides, right? And this has to do with the fact that these water molecules will cluster around solutes like sugar, like this, and they basically get bound up. And so what osmosis is really dealing with is the effective concentration of free water molecules. The more solute you have, the less free water molecules you have, right? And so that drives the direction of diffusion of water molecules in this kind of an experiment. And this is also true in cells. And so these are just some definitions from your book. And probably most important is really just to know hypertonic and hypotonic, what those mean. So, but we can start out with tonicity. So this is the ability of a solution to cause a cell to gain or lose water, right? So that is effectively, you can think of it like, the higher concentration of salts and solutes present in that solution, the more it's going to tend to draw water out of a cell, right, if it's sitting on the outside, for example. If we have an isotonic solution, this means that the salt concentration is effectively the same as what's found inside the cell. There's going to be no net water movement across the membrane in an isotonic solution. And so if you're working with a sample of cells in the laboratory, this would be very important to think about, right? You probably wouldn't want to dump these cells into into water with no salts dissolved in it. And you also might not want to dump the cells into a solution that had 1 molar sodium chloride in it, right? Because both of those would be solutions that would not be isotonic. You would like to try to mimic as closely as possible in the solution that you're using for dealing with those tissues. You'd like to use a solution that is mimicking the solute concentrations that are found inside those cells. Okay, so if we have a hypertonic solution, this means that the solute or salt concentration is greater than what's found inside the cell. The cells in that kind of a solution will tend to lose water. And in contrast, if we have a hypotonic solution, the salt concentration is lower than what's inside the cell, and cells will tend to gain water. Okay, so what does this all mean? Why do we care? This is kind of a nice example, a little illustration from the book, and shows different behaviors of animal cells and plant cells. And the reason for that is that, as you remember from last time, animal cells have a plasma membrane around the surface of the cell, but that's it, right? They might have extracellular matrix, but they don't have what plant cells have, which is a cell wall, this additional layer around the outside of the cell that is even more impenetrable to water. So it makes those cells much stiffer and more resistant to the in and outward flow of water across the membrane. So if we take animal cells and we put them into a hypotonic solution, so again, that means that there is tends to be lower concentration of salt outside in the solution than what we find inside of the cell. That will mean that water will tend to want to enter the cell, right? It will want to go into the cell where salts are more concentrated, and that leads to these cells bursting. They will lyse. And in fact, this is a technique that, if you're working in a biochemistry lab, we actually use this principle frequently if we want to purposely burst cells. So if we take a tissue sample or a sample of cells and we want to break them open to purify the molecules that are inside, we will typically take the cells and put them in a hypotonic solution purposely so that they will lyse due to just this osmotic action of water. If these cells are in an isotonic solution where the salt concentrations are roughly the same inside and outside of the cell, water will flow in and out at roughly the same rate, and these cells will look normal. And then over here, if we have these cells in a hypertonic solution, where we have a lot more salt outside of the cell than inside, now water will tend to flow out of the cell, and these will just kind of shrivel up. Plant cells behave a bit differently, and you can see that sort of illustrated here. They actually prefer to be in a hypotonic solution in general, where there's a lower concentration of salts outside of the cell, which tends to mean that water is in general flowing into the cell rather than out of the cell. Why is that? Well, they've got this cell wall, and if there's water pressure coming in, the cell wall is very strong. The cells don't lyse, but instead they have this sort of turgor pressure. They are turgid. They are stiff, and it means that the cells are that this is the optimal state that they prefer to be in, probably because plants normally have to interact a lot with environments that are aqueous when they're growing, right? They have to deal with dew, they have to deal with rain. There's a lot of times when these cells have water sitting on their surface, and so they've actually adapted so that they actually prefer this situation. If you put these cells in an isotonic solution, they become flaccid. Water is flowing in and out, but they don't have this sort of stiff structure to them. And if we put them in a hypertonic solution, then of course the same thing happens. They tend to shrivel up. So you might wonder, when you go to the grocery store and you go to buy vegetables, and they have the water spray coming on, and they're spraying the vegetables a lot. Actually, that's in part because of this effect, right? You want to have water on the surface of those vegetables. The plant cells, they will tend to be taking up water, and they will look better, and they will be more crisp if they are in that kind of environment, and that's due to this effect. Okay, so transport proteins. So these are proteins that allow hydrophilic substances to go across the membrane. So because, as we said, many molecules wouldn't have the ability, or at least certainly not quickly, to cross the plasma membrane unless they were helped by proteins. And some of these are channel proteins. They have a hydrophilic central pore that allows molecules or ions to go across, like that beautiful example of the potassium channel. There also are proteins called aquaporins that are specifically pores that allow water molecules to go across the cell. And then we have to define a few more things here. So there's a process that's called facilitated diffusion, and this is really just passive transport that's helped by proteins. And so what this means is that some proteins will set up a situation where they allow small molecules to go across the membrane, but the driving force for that transport is just passive diffusion. In other words, these molecules will tend to travel along their concentration gradient. If there's a higher concentration outside the cell than inside, they'll tend to go in, and if there's a higher concentration inside, they'll tend to go out. Okay, and so these are really just corridors that provide for these molecules to get across the membrane efficiently. These would include these aquaporins that allowed water to get in and out. They also include ion channels that can open or close in response to a particular stimulus, and those are called gated channels. So here's a couple of examples. So this is here's a channel protein that allows small molecules to go across the plasma membrane like this. And in this example here, this is just it's a fixed channel. It's sitting there in the membrane, and as we said, these molecules can go across just according to the concentration gradient that exists across the cell. And this is a second example down here, a carrier protein. So these are proteins that can, that again, sit in the middle of the membrane, and they have ability to bind to these various kinds of solute molecules that are, in this example, are outside of the cell, bind to them, and then change conformation and release them inside the cell. Okay, and so those would be proteins that would have very specific ways of interacting with those, with the whatever they're allowing to cross the cell. So a great example again is the potassium channel, where the beautiful crystallographic work has showed that if you look at the structure of that channel, it's set up so that only potassium ions can get through there, right? Not water, not other kinds of ions, but only potassium, and that's because there are very specific interactions that look at the size of the ion and its ability to coordinate with various functional groups going across that channel that makes it incredibly specific for potassium. So, you know, beautiful example of exquisite specificity in biology. Okay, so in contrast to passive transport, we also have active transport going on. And what this means is that there is the application of energy to move solutes against their gradients. So if we have a higher concentration of salts or protons or what have you outside of the cell than inside, if the cell wants to change that, it has to use active transport. It has to use energy to bring those molecules against their concentration gradient into the cell. And that's shown right here. Some transport proteins will use passive diffusion, but many of them will drive solutes against their concentration gradients. This uses energy, usually in the form of ATP, and it's performed again by specific kinds of proteins. Proteins that are passive diffusion channels are not active transporters, right? And vice versa. So they're definitely different, sort of distinct classes of proteins and functions. And this is a slide that shows an example of the sodium-potassium pump. So here's it's really kind of a very interesting, it's sort of like a machine, you know. This is a protein that will bind to 3 sodium molecules that are and has a very high affinity for sodium, because normally sodium on the inside of the cell is at fairly low concentration, whereas potassium ions are at fairly high concentration. So this protein, if it wants to pump these out of the cell and go against this concentration gradient, it actually has to have a pretty high affinity for these sodium ions. And then once the sodium ions bind, there's a change in the structure of the protein that favors a phosphate getting added to it from an ATP molecule. And when that phosphate is added to the protein, it triggers a conformational change, a structural change in the protein, such that these sodium ions are now released. The affinity goes down, and these are released out into the exterior matrix of the cell. And once they're released, that leads to a higher binding affinity for potassium ions that, remember, outside of the cell tend to be at fairly low concentration. So they have to be bound up by this higher affinity binding interaction with the protein. And in this case, 2 potassiums are bound, not 3. So 3 sodiums were released, but 2 potassiums are bound. And once that occurs, that triggers that disfavors this phosphorylation of the protein. So the phosphate gets clipped off, and that then triggers a further conformational change that allows the protein to open up to the inside of the cell. These potassium ions fall off over here, and then the cycle can start again. So it's a really beautiful, this is called a sodium-potassium pump. So this cycle is going on. And there's a couple of very interesting things to note about this. So How do we know all of these details about how it works? Well, again, a lot of this comes from studying the structures of these kinds of pumps, these molecules, and how they work, how they bind to ions, and how they change when phosphorylation occurs. And also, you can see that this is an interesting situation where for every 3 charges that we're pumping out, we're only pumping in 2 charges here with these potassiums, right? So it's going to set up an electrochemical gradient in the cell, which it does. And so this is actually, your book defines this as an electrogenic pump. Electrogenic. It's on one of the later slides. We're coming to it. And so these are ion pumps that maintain something that's important called membrane potential. So membrane potential is the difference in voltage that occurs across a membrane. And this is created by differences in the numbers of positive and negative ions. And you'll see in your book that your book uses the word cation and anion. Hopefully that's familiar to all of you. You know that a cation, hopefully, is a positive ion, right? And an anion is a negative ion. And so if there are differences in the numbers of those across the cell, that will set up effectively a gradient. And it's called an electrochemical gradient because it's both, consists both of the ion concentration across the gradient, that's the chemical force, and the effect on the membrane potential of the effect of the membrane potential on the movement of these ions, which is an electrical force, right? So there's more, if there are more positive charges than negative charges on one side of the cell than the other, that sets up an electrical gradient, right, in addition to a chemical gradient. Right, and so as we said, these electrogenic pumps are proteins that will generate voltage across a membrane like the sodium-potassium pump example that we just talked about. This is the major electrogenic pump of animal cells. In plants and fungi and bacteria, the main electrogenic pump is a proton pump, where protons are being actively moved across the membrane. And this is just a really nice picture that shows how sometimes coupling of this kind of electrogenic pump, such as this proton pump, is important for the transport of other kinds of molecules. So in this example, we have the cell is actually using the proton pump, which is pumping protons actively out of the cell to help import sugar molecules. And that reason for that is that the sucrose transporter is a transporter that works through the import, in part, of these protons. And so protons get pumped out of the cell, and then they can come back in by interacting with the sucrose transporter, and every time they do, they trigger the entry of a sugar molecule. So this is a gradient that's set up to help pump sugar actively into the cell. Okay, and then finally, I want to just define another couple of terms here. Sorry for all the definitions. A lot of biology, biology is kind of a language, right? We sort of have to get a lot of the terms down that we're going to use to talk about these processes. And the last two that I really want you to know about today are the terms exocytosis and endocytosis. So we know that small molecules and water can enter or leave the cell through the bilayer, either just directly, so if they're oxygen or CO2, for example, they'll just diffuse across without any help. If they're ions or water, they go across through these channels that we've been talking about, or sometimes they're pumped across if they're protons. But if they're big molecules like polysaccharides or proteins, these actually have to go across the membrane in vesicles. And this requires energy. And there's 2 different ways that this can occur. There's in a process called exocytosis. These are transport vesicles that come from inside the cell. They migrate to the cell membrane, fuse with it, and then release their contents. And in the reverse process, endocytosis, the cell will actually take in macromolecules by forming a vesicle around it by pinching off a piece of the plasma membrane. And there's a picture here, this is again from your book, that shows 3 different types of endocytosis called phagocytosis, where the cell is effectively eating a macromolecule by swallowing it up into a vesicle and then digesting it, or pinocytosis, where the cell is taking up solute molecules in these much smaller vesicles here and then releasing them in the cell, or receptor-mediated endocytosis, where the cell has receptors on its surface that bind to a particular kind of molecule, and these then pinch off and enter the cell that way. So you can have a look at that in more detail in your book, and the last slide just summarizes these different processes. Have a nice weekend. See you guys on Monday.